Hi, everybody. Thank you for coming. I'm Sarah Buense, and I will be hosting this room for Bears 2020. This year, our Bears looks a little different because of the whole pandemic going on, but we found a way to accommodate, and there are two other rooms that you can also join and see if you'd like to go. If you'd like to go visit those rooms while we have the those presentations going on. We're going to be showing those three Zoom sessions, and feel free to browse around. And here in this room, we're going to have a total of 6 sessions, and we're going to have, after each video, a 2-minute questionnaire. And if you'd like to ask questions and we don't have time after those 2 minutes, then if there's time at the end, we can go ahead and ask questions then. So I'm going to share the first video. Let me go ahead and share my screen.
Hello, my name is Annabelle Verghese, and today I will be discussing the prevalence of horizontal gene transfer within microorganisms of the human microbiome. So a brief summary of the horizontal gene transfer: it's the transmission of genetic material between organisms aside from the means of parent to offspring. So in the process of transformation, the DNA from a dead bacterium is obtained by the beneficiary. And then another method is through conjugation, in which a structure that extends from one organism to another, which is called a pilus, enables them to exchange DNA. And then transduction is the movement of bacterial DNA through a bacteriophage. So within humans, there exists a microbiome that consists of many different microorganisms that are necessary to carry out numerous functions within the body, such as immunity and digestion. And just like prokaryotes that can participate in horizontal gene transfer outside of humans, so can the organisms within the microbiome. So this can actually be very helpful because this process would allow greater amounts of host microorganisms to be able to carry out these vital human functions. And it has also been studied that there's an increase in likelihood of horizontal gene transfer occurring among microorganisms with similar phylogenetic structures, as they're likely to populate similar regions within the body. And there's also a certain bacterium called Bacteroides plebius that is found in the gut microbiome of natives of Japan. And this bacterium actually contains genes that assist in digesting the carbohydrates of seaweed, which is a common part of the Japanese diet. And scientists have found that the genes that allowed this bacteria to do so originated from the marine Bacteroidetes phylum, which indicates the horizontal gene transfer among natural human microbiome organisms and introduced bacteria. And dairy products are also known to have an abundance of microorganisms that, when consumed, may also have the ability to take part in this gene transfer. So aside from these advantages of horizontal gene transfer within the human body, there are also some concerns that arise. So many of the microorganisms within the microbiome have the genes for antibiotic resistance, which is why these helpful bacteria aren't harmed when an antibiotic is utilized to fight off an illness. But these same bacteria can transfer those genes to pathogenic bacteria that enter your body, and this ultimately adds on to the issue in bacterial infection treatment, where antibiotics are becoming less and less effective due to the bacteria's evolving nature. And there is also the concern that harmless microorganisms in the microbiome may become pathogenic through this gene transfer, but there surprisingly haven't been any reports of this situation occurring yet. However, in the case of genetic transfer to a somatic cell, scientists have also examined certain leukemia and stomach adenocarcinoma cells, and they appear to have sequences originating from bacteria in the human microbiome. So this actually suggests a possible harmful relationship between your own microbiome organisms and your body cells, which can potentially instigate further research on the sources of cancer and maybe even treatments or preventions when understanding the correlation between the different human body cells and microbiome organisms. And that is all. Thank you for listening to my presentation on the various aspects of horizontal gene transfer among microorganisms within the human body.
Thanks, Annabelle, for the presentation. Is there anything that you'd like to comment on before we go on to the questionnaire part? Maybe something that you'd like our audience to know about your research?
The only thing is, in my presentation, for the bacterium that is in Japanese natives that can help digest seaweed, I only mentioned the phylum name in my presentation, but the actual genus and species is Zobelia galactonivorus. Just wanted to put that out there.
Okay, great. And then now we can move on to the question part of the presentation. So does anybody have any questions for Annabelle?
What do you think was the hardest part of your research for your topic specifically?
So when we learned about horizontal gene transfer in bio, we mainly associated it with prokaryotes. And like I mentioned in the beginning of my presentation, and how they can transfer genes through transduction or transformation or conjugation. And so when I learned about, when I was doing my research, I learned about how the microbes can actually share it with our own somatic cells. So trying to research how they did that and how they were able to transfer genes with somatic cells, because our somatic cells aren't prokaryotic cells. So that was kind of difficult because they can't necessarily do so in the same ways as other prokaryotes.
Okay, thank you so much.
Question.
Yes.
Hi, Annabelle. Nicely done.
Thank you.
My question is about kind of the microbiome itself, and especially the gut microbiome. Did you read much about that, number one? And number two, if these bacteria are detrimental, like you say, how do you think that we can counteract that or treat for that?
So your first question was about gut microbiome?
Mm-hmm.
What exactly about it?
Well, we learned a lot about the gut microbiome, didn't we, in Gen Bio, know? Right. And so we know that most of the reason that most of the interactions are positive interactions, right? Right. So I was worried about those negative interactions you were talking about.
Oh, so for the negative interactions, people essentially for the gut microbiome, as far as negative interactions in the gut microbiome, I haven't learned a lot of research about that as far as negative interactions go. But for possible cancer-related issues, it's difficult for them to actually trace back exactly that it's because of the horizontal gene transfer that's why the cancer has arisen. But that's what they're working on right now, trying to see how they can use this information in order to try to prevent that from happening.
Thank you.
Thank you.
Did anybody have any questions? Thank you, Dr. Rozelle, for chiming in.
I have one more question.
Okay, go ahead, Kim.
Does everyone who can digest seaweed have that bacterium in their gut?
Not necessarily. So I found that most Americans actually don't have this bacterium. And so if you are American and you can digest the seaweed, there's actually enzymes that you have that are similar to that of the marine bacterium. So not necessarily.
Okay, well, that is the time allotted for Annabelle. And if anybody else has any questions and would like to know more, if we have time at the end, we can go ahead and ask those questions. So now moving on to our next presentation. And this will be for Francis Hune. Sorry if I'm mispronouncing that, but let me go ahead and share my screen.
Hello, my name is Francis Huynh.
My name is Jonathan Nguyen, and in this video, we will be going into more depth about birth defects caused by non-disjunction, a topic that we thought was interesting because of the effects it has on modern medicine and on our everyday human interactions.
Since we are talking about birth defects, we have to mention the process of meiosis, as it is how we give birth and conceive future generations. In meiosis, we end with 4 gametes after undergoing 2 phases of prophase, metaphase, anaphase, and telophase, as shown in the diagram on the left. Today, we will focus on non-disjunction, the failure of one or more pairs of homologous chromosomes or sister chromatids to separate, resulting in an abnormal distribution of chromosomes in the daughter nuclei, which occurs in anaphase. In the middle diagram, it shows that non-disjunction occurs in anaphase 1, resulting in an uneven distribution of homologous chromosomes, which results in one cell containing more sister chromatids than the other. The diagram shows that the cell containing more sister chromatids undergoing metaphase 2 will result in 2 gametes having a ploidy number of n+1. The cell containing less sister chromatids will end with 2 gametes with a ploidy number of n-1. In the next photo, non-disjunction is occurring in anaphase 2. One of the cells did not separate their sister chromatids at all, and it resulted in a gamete that has a ploidy number of n+1 and the other gamete to be a ploidy number of n-1. While the other cell normally underwent anaphase 2 and resulted with 2 gametes with a ploidy number of n. An example of this failure in non-disjunction could result in trisomy 18, which is also called Edwards syndrome. People with Edwards syndrome have a small head and their ears are usually on the lower end of their face. Babies who have trisomy 18 have a 12% chance of living through their first year because they have heart defects and trouble feeding.
Trisomy 13, also known as Patau syndrome, is a severe birth defect since the symptoms include heart problems, brain or spinal abnormalities, and inferior exterior extra limbs. These individuals also experience severe intellectual disability. These cases aren't known to be genetically inherited and rather result from a random error during the formation of eggs or sperm in healthy parents. There is a 20% chance that the babies born with trisomy 13 survive their first year of life. Next, we have trisomy 21, also known as Down syndrome, which is a condition involving the addition of an extra copy of chromosome 21 in an individual. This has many physiological effects on the person with this condition, involving low muscle tone, upward slants in the eyes, small stature, flattened face, and heart conditions. Trisomy 21 is described as a developmental intellectual disability as well, and causes these people to learn skills at a slower pace compared to those individuals who do not have it. Unlike trisomy 18 and trisomy 13, though, these individuals with trisomy 21 are able to live past their childhood and can live a full life.
Next, we'll be talking about Turner syndrome, which is a genetic disorder that affects the sex genes of females, involving the X chromosomes either being partially missing or missing completely. Turner syndrome can be detected prenatally and involve physiological changes including low set ears, cardiac defects, small jaw, shorter stature, and many more. This disorder can be especially troublesome because it can impair motor movement development as well as cause heart, hearing, and vision problems.
Lastly, we have Klinefelter syndrome, which is a genetic disorder that affects the sex genes of males, involving the addition of an extra X chromosome. This genetic disorder can only happen to males and involves physiological changes / hormone imbalances. These changes include lower production of testosterone, development of breasts, and smaller testes, as seen on the diagram to the left. Many males who have Klinefelters may also have slow motor development as a baby. This disorder also has a possibility of making the person infertile, one of the most common effects of Klinefelters. Next, we have our references, and thank you so much for listening to our talk.
Okay, well, good job, Jonathan. And is there anything that y'all would like to share about your your project or presentation?
I don't think so. We can get to the Q&A.
Okay. Does anybody have any questions?
Yeah, I do. What is the average life expectancy of people with the condition of trisomy 18?
Well, usually they only live to like the first year. So when they're like 1 year old, that's usually the latest. But there's actually been a couple of cases that we did a little bit of research on, in which there was a person that is in their like late 40s that has trisomy 18. But again, when Francis was listing off all of those physiological issues that these people with trisomy 18 have, it's very difficult for them to live past that first year. And it could also be due to the fact that they had the other form of the disease, which unfortunately we didn't do too much research on. And it's basically the mosaic form of trisomy 18. But again, we just talked more about the non-disjunction form of trisomy 18. And that was sort of like the scope that we kept it in. So yeah.
I think Cecilia just asked a question. She said, what surprised you the most from your research?
I think for me, it was that, I got to learn more about trisomy 18 and trisomy 13, and they were diseases that I never really knew about, I guess. And the fact that they're very influential in like development, in the fact that like you cannot, it is very, you sort of need like a miracle, I guess, in order to live past that first age. And I guess it sort of raises questions to like how modern medicine could possibly fix something like a disease such as this. And I just thought it was very interesting that, again, these people can only live usually to like throughout their like first year of life. And then after that, usually they die after that. I just thought that was very interesting that there was a disease that kind of caused that sort of issue. But I don't know about Francis.
Well, for me, I thought Turner syndrome and Klinefelter syndrome was pretty surprising for me, since like it only affects the respective genders. Like how Turner syndrome only affects the sex gene of females, and like how Klinefelter syndrome only affects the sex genes of males, and like how it makes the person infertile and everything.
Okay, yeah. I also found it very interesting how, non-disjunction can affect all of those factors. So, yes, good job, you guys. And does anybody else have any quick questions before we move on to our next presentation? Oh, and also I shared the flyer with you guys. So if you'd like to move back and forth from different rooms, the link is on there. It's hard coded onto our flyer. So go ahead and, check those out if you'd like. So I'm going to move on to our next presentation, and this is telomere attrition and its effects on stem cells. And Cody Mai is our leader for that one. So let me go ahead and share that with y'all so you can see it.
Hello, my name is Cody Mai, and today I will be presenting on the concept of telomere attrition and its effect on stem cells. So the first thing I want to talk about is aging. And aging can be summed up as this progressive decline of tissue function that eventually results in our mortality. There are many factors that affect aging, including our environment, but there are cases that provide evidence that there is a genetic component within our genome that is responsible for aging. And we can see that by looking at these next two images. In the second image, this individual does not seem to age. You can see that as she grows older, she physically looks the same. And what's interesting is that her brain structure and neuroendocrine functions are all infantile, and yet she is 15 years old. Findings suggest that she suffers from developmental disorganization resulting from a mutation of a regulator of development. And in this third image, we can see an individual with Werner syndrome, which is a hereditary condition associated with premature aging. And so by looking at this published data of people who have or who are born with a certain predisposition to age faster or slower, these case studies help identify developmental genes responsible for aging. And that genetic component has been found to be telomeres. So what are telomeres? Well, in our cells, chromosomes make up our DNA, and at the end of every chromosome, there are telomeres. Telomeres are made up of these repeating sequences of DNA, and at the end of each telomere, there's a 3' overhang that forms a T-loop, which establishes a capping structure, and this structure protects chromosomes from exonucleases, DNA repair mechanisms, and recombination. And throughout life, cells multiply, right? They accumulate these cycles of division and replication in order to renew damaged cells and tissue. However, every time replication occurs, the telomere shortens, and they progressively get shorter with time and as we age. An analogy that is commonly used is that when you think of chromosomes as shoelaces, telomeres are the little protective tips at the end. And during our lives, they tend to wear down. And when telomeres can't protect chromosomes properly, cells can't replenish and they malfunction. This sets up for physiological changes in the body, which increase the risk of the major diseases of aging, such as cardiovascular disease, diabetes, cancer, and even a weak immune system. But there is a specialized enzyme known as telomerase, which can add DNA to the ends of chromosomes to slow or prevent and partially reverse the shortening. And so we can see that the length and integrity of telomeres are maintained by this enzyme. So how does the shortening of telomeres affect stem cells? Well, in humans, shortened telomeres are regarded as risk factors and associated with deficient cell proliferation and tissue degeneration. And so critically short telomeres trigger apoptosis, chromosome senescence, and loss of stem cell viability. So what is a stem cell? Well, a stem cell is an undifferentiated cell which can both generate specialized cells such as skin, blood, or neuron cells through cellular differentiation, and they can also remain in the body by proliferating. However, as we age, our stem cell count decreases and we have less and less new somatic cells, which may lead to the decline in organ function. And this is because although telomerase is expressed at very high levels during embryo development, it is reduced in adult stem cells. So the level of telomerase activity is low or undetectable and insufficient to stably maintaining their telomere length. And so this progressive shortening of the telomeres as we age seems to act as a barrier to hematopoietic stem cell development in the bone marrow. And this limits the regeneration of other hematopoietic stem cells and the renewal of all blood cells, which may develop into bone marrow failure, intestinal disorder, or malignancy. And so the critical importance of telomerase activity in human stem cells has been highlighted as the cause of dyskeratosis congenita. In this disorder, a defect of the telomerase RNA template gene results in the absence of telomerase activity, and this causes premature telomere shortening. And since as we know, telomere shortening alters proper stem cell function, this can be visualized in the symptoms of dyskeratosis congenita. So we can see abnormalities of the skin, such as unusual pigmentation, defects in fingernails and toenails, oral lesions that appear in the mouth. And so combined with the shortening of telomeres, and the low presence of telomerase in adult stem cells, these alterations accelerate the aging of mechanisms and trigger stem cell degradation. This is significant as a whole because they would be a great target for therapies based on telomerase regulation and preservation in order to fight against aging. So understanding telomere and telomerase dynamics in stem cells could lead to the development of novel therapies in aging-related diseases and cancers. Thank you.
Good job, Cody. That was a really good presentation. Is there anything that you'd like our audience to know about your presentation or your research that you've done on this topic?
Well, I did this because I was really interested in telomeres and aging and how the shorter our telomeres get, the faster we age. And so that's what initiated my research project.
Okay, awesome. Does anybody have any questions for Cody?
I have 2 questions, if that's all right.
Okay.
First is, what's the clinical spectrum of bone marrow failure in the telomere attrition and the stem cell dynamic?
Okay, well, stem cells regenerate our bodies in a similar manner to match ignition. So in a sense, stem cell ignition has to be precisely tuned to avoid uncontrolled proliferation that occur in tumors, or inversely, the lack of proliferation that happens in degenerative disorders. So specifically, you mentioned bone marrow failure. There have been many cases where a subset of stem cells isolated from adult bone marrow showed shorter telomeres than a similar subset isolated from fetal liver or cord blood, suggesting that there's a progressive decline in telomere length with age that occurs in hematopoietic stem cells. So what's interesting is that it's possible that the transplanted hematopoietic stem cells derived from aged donors may reach their proliferative limit during the lifetime of the recipient. So in fact, there are actually several cases with graft failure after stem cell transplant because they have been reported to have unusual telomere shortening. So hematopoietic dysfunction caused by the defective telomere structure and repair does have a broad clinical spectrum. And there have also been cases where it leads to aplastic anemia, which is a rare condition in which the body stops producing enough new blood cells, which is a result of blood marrow damage. Hopefully that answers your question.
Awesome. The second would be, what kind of cancerous malignancies could telomere attrition lead to?
That's an interesting question because it's believed that cancer occurs because a genetic mutation can trigger the production of the enzyme telomerase, which prevents telomeres from shortening, and so cells can continuously proliferate. But it has been found that shortened telomere length and increased telomerase activity are associated with reduced response to chemotherapy, faster disease progression, and poor prognosis in patients with acute leukemia. So for those of you who don't know, acute leukemia is characterized by the uncontrolled proliferation of myeloid precursor cells or lymphoblast cells. And so the shortened telomere length is observed in patients with leukemia and is associated with causing instability in chromosomes. Hopefully that answers your question.
Okay, good job, Cody. You're doing awesome. Does anybody else have any questions? Okay, so also I wanted to mention when we leave, the flyer that I attached also has a post-assessment form. So if I can have everybody fill that out so you can give us feedback on what you thought about our research symposium. So, okay, that being said, I'm going to move on to the next presentation, and our leader is Kim Pham, and she will be talking about the growing antibiotic resistance epidemic and its effect on treatment plans for respiratory diseases.
Hello, my name is Kim Pham, and today I will be going over a general science interest topic that I was introduced to through my General Biology 1 course with Dr. Vargas. This presentation covers the growing antibiotic resistance epidemic and its effect on treatment plans for respiratory diseases. Antibiotic resistance is whenever microbes, bacteria, and or fungi develop the ability to defeat the drugs designed to kill them, according to the CDC. In this case, this is whenever the antibiotic would only affect the non-resistant strains of the microbes, in which the microbes that do not die are the resistant strains. And these resistant strains would be typically the last ones remaining after a trial of antibiotics. And these resistant microbes would grow and multiply amongst each other, in which some could develop into very dangerous superbugs. In relation to respiratory illness, there is a growing epidemic in the U.S. concerning antibiotic resistance, since there is a growing drug resistance within some of the most common respiratory pathogens worldwide, including strep and tuberculosis. The antibiotic resistance would only cause complications in treatment for many of the most common respiratory illnesses, and this presentation will cover asthma and COPD, also known as chronic obstructive pulmonary disease. In relation to asthma, this presentation covers the AMAZE study, in which the effects of azithromycin are observed. This azithromycin is an antibiotic that treats exacerbations for the severely asthmatic. The results of the AMAZE study show that azithromycin reduced the Haemophilus influenzae load, which is what caused the exacerbation, the asthmatic exacerbation, but also increased resistance to antibiotics. And because of the nature of antibiotics, this antibiotic decreases bacterial diversity within the human microbiome, which already is a factor that would lead to further health complications. COPD, bacterial infection is one of the more serious causes of COPD exacerbations, and in fact, they cause at least half of COPD exacerbations, and they are typically the primary cause of these complications. There is increased mortality that is correlated with a high level penicillin resistance. Penicillin typically is treatment for patients with with only mild COPD and no prior exacerbations, which means that with continued use, this would become essentially ineffective whenever more serious exacerbations would occur. Something to consider is that a diverse microbiome is a large factor in promoting overall physical health within humans, and it is best not to create an imbalance over time. Antibiotics are often one of the primary causes of a microbiome imbalance, which often leaves the human susceptible to all kinds of dangerous pathogens. The amazing study mentioned in regards to asthma highlighted the need for studies assessing the efficacy of non-antibiotic macrolides as a long-term therapy for patients with persistent uncontrolled asthma, while the COPD study depicted that antibiotic treatment should be restricted to COPD patients with more serious illnesses who are at risk for infections with antibiotic-resistant strains. Essentially, both these studies showed parallels of reducing antibiotic use for the sake of preventing more serious complications down the line. Asthma and COPD are often chronic illnesses that an individual has to live with for the rest of their life, and it is best not to put strain on these patients for in actuality making their disease worse because of the antibiotic resistance that would result from the frequent use of antibiotics. These are the sources I referenced throughout my presentation. Thank you so much for listening.
Okay, Kim. Good job on your presentation. Thank you for being a part of BEARS this semester. Is there anything that you would like the audience to know about your research?
Sorry. So I chose this topic specifically because, whenever you think about common respiratory illnesses like asthma or COPD, you would typically assume that treatment consists of like inhalers or nebulizers or albuterol, etc. But my presentation specifically focused on the severe exacerbations of both, because typically the inhalers, etc., are used as prevention for the severe exacerbations of that.
Okay, good job. Does anybody have any questions for Kim?
I have a question. So do you think that the implications that you talked about can also apply to other illnesses that also heavily rely on antibiotics?
Yeah, so pretty much anytime you use antibiotics or you heavily rely on them, you're putting yourself susceptible to antibiotic resistance, which would in turn lead to issues with a diversity within the human microbiome. And so any other illness, that it doesn't necessarily have to be respiratory illness. It can be anything like something that has to do with your digestive system, anything like that. You're putting yourself at risk for making yourself susceptible to potential superbugs. And so I think that pretty much the takeaway from my presentation is just to kind of encourage less reliance, or kind of develop new therapies or treatments that don't heavily rely on the practice of using antibiotics so frequently within healthcare.
Okay. Does anybody else have any questions for Kim? Okay. Well, thank you, Kim, for sharing. Good job. I'm going to move on to our next presentation. And this is over the topic living with HIV in the 21st century. And it will be led by Susana Villazon.
Hello, my name is Susana Villazon, and I'm going to speak about a topic I feel very passionate about, which is living with HIV now versus 4 decades ago. As many as you may recognize, this is Freddie Mercury. He was the lead singer of the band Queen. I was shocked when I found out that he died at age 45 due to a complication of AIDS. After this, I became even more interested in the topic. So what is the origin of HIV? The most accepted theory is that it crossed from chimps to humans back in the 20s in the Republic of Congo. There were some people in the community who used to chase and hunt chimps So what happened is that the virus that was originally in chimps mutated enough to cause disease in humans. But it wasn't until 1981 when the CDC published an article about 5 previously healthy males who had a red infection. More cases like this arised, and in 1982 the CDC uses the terms AIDS to describe the disease that is causing a defect in the cell-mediated immunity. Up until this point, they weren't sure about what was the agent that was causing AIDS. They thought it may be a virus, but weren't sure. Until 1985, when they named it as HIV, as a causating agent of AIDS, which is a later stage of the disease. In order to treat these patients, because there wasn't a lot of information, in 1987, the FDA approved a drug that was 50 years ago used for cancer, Zidovudine, as a treatment for HIV infection. They found out that the drug may stop the replication of virus. Nevertheless, this wasn't enough, and people were still dying because of complication of AIDS, very young people. So, thanks to the effort of the scientific communities, the highly active antiretroviral therapy, HAART, came through the 90s, which is known as the HAART era. Basically, what they planned was that in order to stop the replication of the virus, they have to target in multiple points, meaning that they didn't target just one point about the life cycle of the virus in the human body, but multiple, just as the entry of the virus in the cell, and also the enzyme that this one used. So they knew they have to combine different drug mechanisms in order to diminish the replication of the virus and disease. They also came up in 1997 with a drug called Combivir that what it did is combine 2 pills in one, and this made life easier for patients. Here is some fact that is very important because the Department of the United Nations Design for AIDS claimed that in 1992, AIDS was the number 1 cause for U.S. men between age 25 and 44. 3 years later, and also thanks to the antiretroviral therapy, there was a steep decline in AIDS in the U.S. And here is what amazed me more about the scientific progress that we have done in this disease. In 1996, a patient infected at age 20 had a life expectancy of 19 years, meaning that he or she will die approximately 39. In 2011, 9 years ago, a patient infected at the same age, the life expectancy was about 53 years, meaning that he or she will die approximately 73. This, keeping in mind, is with continuous pharmacological therapy, meaning that the patient has to take the antiretroviral therapy as soon as they know they're infected and don't stop ever the medication. Up until this point, there are some studies that claim that the life expectancy has even increased more and is pretty similar to someone that is not infected with HIV. Nevertheless, it's not just about the number of years that the person had, but the quality of life. That's why, like this study, many lawyers are claiming that we have to target also the comorbidities and the life quality that these patients are living, because it's not just about the years that they're living, but how are they living these years. And I want to end up with who I started, with Freddie Mercury, and I like this quote of him because he said the most important thing is to live a fabulous life, and as long as it's fabulous, I don't care how long it is. Thank you.
Okay, so if she comes back, we can come back to her video or not come back to her video, but ask questions to her later. So we're going to move on to our next presentation, and it's the efficacy of masks. And our leader is George Fakes. So I'm going to go ahead and share that with you guys.
Hello, my name is Jamil Akhtar.
And I am George Fakis, and our project is about the efficacy of masks based on the different materials. The coronavirus has taken the entire world by storm, plaguing every single person's life in one way, shape, or form.
There are over 11 million cases and 250,000 deaths. With the numbers both rising, there must be a way to prevent it.
We know that many people, like the CDC themselves, have promoted the use of masks, but there are so many to choose from. The question remains, which one is the most protective?
We know masks are used to filter different particles, and different filter types are in different masks, which stops particles from penetrating the mask and allowing the particles to get into your body.
The most prevalent masks used by professional surgeons and medical staff in their duties are N95 masks. So our hypothesis is that N95s, followed by surgical grade masks, are the most effective in preventing the penetration of COVID particles.
The COVID virus is very small, with the particle size being 0.1 microns. In comparison, the N95 filter is about 0.3 microns large, so the hole is 3 times bigger than the virus.
Yes, the particles usually attach to something larger though, so the filtration can catch it easier. But it's like trying to toss a ball into 2 targets. It is possible, but it's much more difficult to hit this ball into a bucket compared to like a larger swimming pool. It's much easier when the target is a lot bigger.
After collecting data, we found that the N95 masks filter at least 95% of airborne particles. They are very good at what they do, but are not as readily available to the public compared to other mask types.
When it comes to surgical masks, they are also very effective. The amount of particles it filters ranges from 60 to 80%, and although these are one-time uses, there are plenty available in the US currently.
When it comes to cloth masks, these can range heavily based on how thick the material is or how many layers there are. There were many cases in which the thicker, higher quality mask proved to be just as effective or even more effective than the normal surgical mask, with the percentage of blocked particles ranging from 30 to 82%, though not being nearly as effective in fluid resistance.
And lastly, we have the bandana/neck gaiters, proving to be the lowest performing of the bunch. Data has shown that the material is really too thin to truly provide adequate protection, with it blocking only around 20% of particles, but some protection is at least better than none.
With no mask, droplet and particles from coughing or even speaking can travel up to 26 feet or remain in the air for minutes. So clearly any sort of protective face mask is better than nothing. When it comes to which is best, it is clear that the N95 mask kept its high standards and proved to be the most effective in preventing particles.
The cloth and surgical mask definitely proved to be very reliable when it came to the amount that the mask was able to stop. Not as effective as the N95, but in the quantities available, definitely proved to be a great option in an attempt to help prevent the further spread of COVID.
Lastly, the bandana and neck masks did provide some protection, but it did not block the majority of particles. It was not really seen as reliable or very effective.
In conclusion, yes, the data supports the hypothesis as the material in the N95 filters out 95% of viral particles, followed by the layers in which a cloth mask is composed of. Based on the layers within the mask, its effectiveness can be greater than that of a surgical mask.
Our interpretation of the results is evidently that N95 masks are the most effective masks in terms of filtration and utility, followed by surgical masks and cloth masks, which have varying effectivenesses depending on the material of the layers that compose the mask. Though if the cloth masks are made of chiffon and silk, they will outperform the effectiveness of the surgical mask. However, other materials can yield results that are less effective. Furthermore, the bandana masks are tremendously worse than all other candidates, as they barely reduce the chance of infection as time develops.
And while yes, masks do help to prevent the spread of corona, staying socially distant and avoiding large crowds is also very important. It is better to stay outdoors and avoid unnecessary interactions if possible, but ensure to wear masks and stay distant whenever interacting with others. In comparison to previously published data, it's been made known that the material that a mask is made of has an effect on how well it will defend against viral particles. In addition, it was safe to assume that the N95 would prevail to be the most effective mask. However, our data did differentiate with some sources that believe the cloth masks are less effective than surgical masks.
However, based on the material and layers that compose the mask, a cloth mask can be more effective than a surgical mask. Additionally, it has been made well known that bandana masks are the least effective masks, which in our studies was proven to remain true. Thank you so much for your time, and once again, we are Jamil Akhtar and George Fakas.
And this is how the effectiveness of masks is determined by the material it is composed of.
Okay, George, I see that you're here with us. Is there anything that you'd like our audience to know while you were doing your research on the efficacy of masks? I really enjoyed that. And I kind of myself thought like before watching your video, I was like, "Ooh, I don't know how safe those bandanas are." So it's actually really good to know that, my guess was correct. So
Yeah, for sure. I guess something to like point out is whenever we initially made the slides, there was 10 million coronavirus cases. Whenever we did the audio, there was 11 million. And since recording this video, there's currently 13,970,000. And to show just in the last 2 weeks that we've been able to grow almost 3 to 4 million cases. So masks do work, but it's obviously, like said in the video, it's really important to make sure we follow all guidelines to try to prevent this spread.
Right, exactly. I totally agree. Even with things like holidays that have been coming by, I think that's also one of the reasons why we have such an increase, because everybody's trying to spend time with their families, and it's just safe to social distance. So yes, good job, George. That was a really good presentation. So does anybody have any questions for George or Jamil? Okay, well, so that concludes our Bears presentations. Thank you so much for coming, you guys. I know that this isn't how we usually do things, but like I said at the beginning, we always can find a way for thank you, Dr. Larios. It's really good just to still get together and be able to do things like this, even though we have to kind of adapt. I think that's a really good skill that we all need to have, is learning how to adapt and to the changing circumstances. So yes, I really thank everybody for coming to our Bears event, and I think this year went really great. And I can't wait to see what we do next year. Hopefully we get to see each other in person. So yes, and I also, please don't forget to fill out the post attendance form. This is for us, and so we're able to choose a winner for the contest that we have for Bears this year. So I'm gonna go ahead and send that out one more time just to make sure everybody has that. And I'd like you to have that filled out by this Friday, as we're gonna be choosing a winner on, we're gonna be announcing the winner on Monday. So yes, so Yesenia just asked, was it okay if I only responded to room A? I only stayed in this room. Yes, so if you only stayed in this room, that is completely fine. Thank you for joining us and staying with us. Okay, so it said NA was required for the rest of them. So yeah, you can just go ahead and put NA if you didn't visit any other rooms. But if you did, we'd like to hear your feedback on that as well. And does anybody have any final comments or questions before we log off for the day?
Thank you.
Okay.
Sarah, this is Dr. Larios. Thank you so much for hosting. You guys did beautifully. I floated around some of the rooms, so I didn't get to watch all of it, but the student presentations were great, and I'm proud of all of you for coming together and putting this amazing event together. But since I did miss some of it, are you guys going to make the recordings available?
Yes, I have it recorded, so I'm going to go ahead and send that out to Dr. Amin and Dr. Miller, so they should be getting those posted.
Awesome. Thank you so much.
Okay.
And thank you for everybody that participated. You guys did amazing. Yay.
Okay, does anybody else have any final questions or comments? No? Okay, well, thank you again for joining us. We hope to see you next year, hopefully in person at our research symposium. And again, my name is Sarah Ponce, and thank you guys for coming. Bye.